For our next assignment, we're going to be modeling Mendel's laws. Gregor Mendel is a person who lived during the 1800s, and we're going to be looking at the history of genetics through the perspective of Gregor Mendel. So the first thing that you will need to do for this assignment is to open your online textbook and read Mendel's discoveries on page 172 to 174. So I'm going to click there now and take a look at who Gregor Mendel was. Now, you will need to read this whole reading selection, but I'm just going to hit a few of the highlights in this video. But Gregor Mendel, like I said, was a man who lived during the 1800s, and as an adult, he was a monk who lived in a monastery in Austria. And while he lived in the monastery, he was put in charge of the gardens. And he grew all the different food crops that fed all of the monks that lived in the garden, and probably also some of the plants that they grew helped to feed the community around where the monastery was in Austria. But Gregor Mendel started out with his curiosity about how living things pass on traits, because he noticed as he grew pea plants and other plants that the offspring plants that came from the seeds of the original plants that he had planted, sometimes they had similar characteristics to the parent plant, and sometimes they had different characteristics than the parent plant. And so this began with his curiosity, the study of genetics. And sometimes Gregor Mendel is actually called the father of genetics because of his studies of hereditary traits. And here's just a few examples of some of the traits that Mendel studied with pea plants. As you see on this chart, which is in your book in the reading passage, there's differences in the plant's traits, and these are just with the pea plants. Plant height can be different. Some plants are tall and some plants were short. The flower position, sometimes the flowers grew along the stem, and sometimes the flowers only grew at the tips of the stem. Sometimes the flowers were purple, sometimes they were white. Sometimes the pea pods were smooth, and sometimes they were bumpy. Sometimes the pods were green, sometimes they were yellow. Sometimes the seeds were round and smooth, and sometimes they looked wrinkled. Sometimes the seeds were yellow, and sometimes the seeds were green. And so all of these were just differences in genetic traits that Gregor Mendel saw and recorded about the pea plants. And so through his experiences and experiments, we have a lot of the knowledge about genetics that we still use today. And so if you'll read that reading passage, you'll get a lot of background information about Gregor Mendel. Okay, the rest of the assignment says for you to watch this video, and in this video I'm going to talk a little bit more about Gregor Mendel in just a moment and about Mendel's laws. And you will use the information from the reading passage and from this video to help you fill in your graphic organizer. And your graphic organizer looks like this. Okay, Gregor Mendel has 3 laws of genetics that we still recognize today. They are scientific laws. And so those laws are the law of dominance, the law of segregation, and the law of independent assortment. So in the reading passage I'm about to show you in this video, you're going to take notice of the law of dominance and state that law here. And then we will read about the law of segregation. You will state it here. And then the law of independent assortment you will state here. So for all 3 of these, you will write down what the law says, and then also we will show an example of these laws. Okay, so I'm going to go to this information about Gregor Mendel. I'm going to kind of scan through and read some of this to you since you do not have access to this in your textbook. This is some extra material from Hudson Alpha. So again, we said Gregor Mendel was a monk who lived in the 1800s, and he studied how physical traits are passed from generation to generation. And like I said, he's sometimes referred to as the father of genetics because he was one of the first people to identify the important characteristics of the genetics process. And most often when we talk about Gregor Mendel, so are his pea plants, because that was the primary plant that Gregor Mendel studied in his monastery garden. And also, we use Punnett squares. Punnett squares are a modern way to show how traits are inherited, and tomorrow later this week, we will be using Punnett squares and learn how to use those to help us to predict genetic traits. So Mendel's work led him to develop 3 major laws, and each of these laws are the basis in the biological processes of meiosis. If you remember us studying about meiosis, meiosis is where the egg cells or sperm cells are created. The reproductive cells are produced through meiosis. Okay, and later this week we'll also be focusing on these 2 words again, the words genotype and phenotype. And right now, you just need a basic understanding of these 2 words. Phenotype is the organism's outward appearance. So, like for you, for example, if we were talking about your hair color or your eye color or something like that, that is a physical trait that we can actually see by looking at you, then that would be your phenotype. Genotype, though, is your actual genetic contents. That means what actual traits are in your DNA. What traits did you inherit from your mother and father? And so we write genotypes as pairs of letters. Capital letters represent dominant traits, and dominant traits show up more often. Lowercase letters are used to represent what we call recessive traits, and recessive traits show up usually less often. Recessive traits can be covered up by dominant traits. Like, for example, in humans, sorry about that interruption. Like for humans, for example, brown eye color is a dominant trait. And so if you inherit brown eye color from either of your parents, then you most likely will have brown eyes as your phenotype. If you have blue eyes, then that is the result of a recessive trait. Blue eyes show up less often than brown eyes. And if one of your parents has brown eyes and one have blue eyes, then the majority of the time you're going to inherit that dominant trait. So you would most likely have brown eyes if at least one of your parents have brown eyes. But your parents can also be what we call heterozygous for their eye color. So you might have actually inherited a different color eye trait from a brown-eyed parent. So anyway, we'll talk more about that as we go through the lessons this week. So if you will bear with me for a second while I scroll down, we're going to come to our first law, one of Mendel's laws. If you need time to type this in on your graphic organizer, then you might want to pause the video while you type in this law. But it's Mendel's first law, the law of dominance. It states that some alleles are dominant and others are recessive. An organism with at least one dominant allele for a gene coding for a particular trait will exhibit the dominant form of the trait. An organism will only express the recessive trait if no dominant alleles are present. And again, that would be like the eye color example I used a moment ago. If you inherited 2 dominant alleles, then you would definitely have brown eyes. If you inherited 2 alleles for blue eyes, then you would definitely have blue eyes. But if you inherited 1 allele for brown eyes and 1 allele for blue eyes, then the dominant trait, brown, would be the one that would show up. So to recap, an allele that behaves in a dominant manner requires only one copy to determine the trait, regardless of what other allele is present. Recessive alleles must be present at both copies for the trait to be seen. If a recessive allele is only at a single copy, its impact will be hidden by the dominant allele. Okay, and then let's scroll on down till we see the next one of Mendel's laws, and that is Mendel's law of segregation. And you see that right here. You might want to pause the video so that you will have time to type that in on your graphic organizer. But Mendel's law of segregation states that during meiosis, the alleles for each gene separate from each other so that each gamete, the egg or the pollen, if you're talking about plants, instead of pollen, it would be sperm if you were talking about animals. Each gamete, egg or sperm, carries only one allele or one version for each gene. Like in this example here, they're actually showing several different traits together here. They're showing the traits for whatever they're using F as the letter to stand for, B and N here. But let's just use F as our example. Okay, this is an example of a diploid cell. So this would be like a regular cell that contains the full amount of genetic information for an organism. If we were talking about humans, this would be one example of a human cell. And let's say that the capital F stands for, let's just use something pretty common here. Let's say that the capital F stands for freckles, and the lower, or actually, capital F stands for no freckles, and the little f stands for freckles. Okay, so this particular person here has the allele for freckle or no freckles and an allele for freckles. Since no freckles is dominant, then the person would not show any freckles. Okay, so if we follow that trait through meiosis, if this cell was going to be copied and go through meiosis to produce egg cells or sperm cells, then this allele right here would be split into this half during meiosis 1. This allele would follow this path and be in this cell in meiosis 1. Okay, then in meiosis 2, this allele would break apart, so we'd have capital F here, because that allele split in 2. We would have lowercase f here and lowercase f here because that allele split in 2. So out of this person's egg cells, let's say this was a female, out of their egg cells, then 2 out of the 4 egg cells would have the capital F gene for no freckles, and 2 of the egg cells from this parent would carry the lowercase f, which means for freckles. So in this case, this parent would have a 50-50 chance of passing on freckles or not. So Mendel's law of segregation again states that the alleles get separated from each other during meiosis. Okay, let's move along. We just have a couple of minutes left for our video. So let's move along to Mendel's law of independent assortment. Mendel's law of independent assortment, again, if you need to pause here so you can type it in on your graphic organizer, it states that genes for different traits can segregate independently during the formation of gametes. So what this means is that these genes don't stick together as they go through meiosis. Okay, so again, if we scroll up to this meiosis example, you can see that, well, in this case, we don't have any of the heterozygous, but you can see down here that in this combination, we have big F, little n, big B from those traits. And over here in these two gamete examples, we have little f, big N, big B, or I'm sorry, little b. So what this law of independent assortment says is that just because you have the dominant trait for, let's say, no freckles, doesn't mean that you will have the dominant trait for brown eyes. Or just because you have the dominant trait for no freckles doesn't mean that you would have a short-shaped nose. So all of the different genes for traits separate or segregate independently of each other.